The topic of the discussion here is organizer concept and induction process. The organizer is an embryonic tissue which organizes the surrounding tissues to develop an embryo. The existence of the organizer was discovered by Spearman. He was given Nobel Prize in 1935 for his landmark discovery. Transplantation is a process in which small piece of a blastula, gastrula or even an embryo is cut off and inserted into suitably prepared incision of the same or another embryo. The embryo from which a part of tissue is taken is referred to as the donor, and the embryo to which the part is transplanted is called the host. The transplanted tissue is known as the graft. Starting with Spearman's experiment and the embryonic induction. Embryologist Hans Spearman succeeded in scratching out the cells of the dorsal lip of the blastopore of an early gastrula of a newt Triturus cristatus before their inward migration. Then he transplanted this small piece of tissue near the lateral lip of the blastopore of another newt. He studied that the transplanted piece of dorsal lip invaginated and developed into a second notochord and the somites. It induced the host ectoderm above the location of the graft to form a neural groove. This neural groove was made up totally of host cells. It underwent normal development, forming a neural tube at first and then a central nervous system. At last, a two-headed creature was formed. Thus, the dorsal blastopore lip of the blastula had the ability to induce the formation of the neural plate in the ectoderm of the host. This phenomenon is called neural induction. The presence of a notochord, kidney tubules, and an additional lumen in the endoderm corresponds to gut of the secondary embryo. All these parts would not have developed if the transplant had not been present. Spearman concluded that the transplanted cells had changed the normal path of development of the host cells. The host cells above the graft were inspired to create a second central nervous system. This influence of one structure in the formation of another structure is called as embryonic induction. The structure which induces the formation of another structure is called inductor or organizer. The chemical substance that is emitted by an inductor is called an evocator. The tissue on which an inductor acts is called the responsive tissue. Now we will learn about the primary, secondary, tertiary and quaternary organizer. Starting with primary organizer. When Spearman transplanted portions, only graft taken from the dorsal lip of the blastopore and the adjoining parts of the marginal zone were found to be able to induce. It is the capability of the dorsal lip of the blastopore when transplanted to cause the production of a total embryo that Spearman called organizer. Spearman gave this dynamic region the name of organizer because of its significance in organizing the development. He imagined the organizer initiating the development of the morphogenesis and differentiation by inducing the structure of the neural tube. Thus, the dorsal lip of the blastopore is termed as the primary organizer. Moving ahead, we will now learn secondary, tertiary and quaternary organizers. As gastrulation continues, the different organ system of the embryo are laid down under the power of the primary organizer, and they themselves then obtain the influence of inducing later formed structures to develop. It is thus probable to know a sequence of secondary, tertiary and quaternary organizers which are set in a sort of chain of command at whose summit is the primary organizer. These developed tissues then work together with another tissue in rotation and induce it to develop. In other words, one tissue gives the stimulus for the development of the other tissue subsequently. Now, we will talk about the structure of organizer. The organizer has a special region. Each region has the capacity to induce the development of a specific organ. There are two very important regions, that is, head inductor and trunk inductor. Head inductor. It is the anterior part of the chordomesodome because it induces the development of head organs. The head inductors may be additionally divided into the archencephalic inductor, which induces the prosencephalon, eyes and the nose rudiments, and the deuterencephalic inductor, which induces the hind brain and the ear vesicles. Trunk inductor. The later part of the chordomesodome functions as the trunk inductor. It induces the development of trunk organs and the tail bird. The neural tube under the induction arises from the chordomesodome. The endoderm and the mesoderm cannot develop into the neural tube under this stimulus. Therefore, the ectoderm has the competence to develop into the neural tube, but the endoderm and the mesoderm do not have the competence to develop into the neural tube. Thus, hereby we will talk about the competence. Weddington in 1932 introduced the concept of competence. It is always related to particular stimuli and particular corresponding responses. Competence is a term which sums up the ability of the enzyme complement of the embryonic cell to adapt to a particular ratio of metabolites. When the ectoderm of amphibian embryo is transplanted from various developmental stages of blastula to early neurula, it gradually loses neural competence. With aging, the ectoderm gradually loses its capacity to respond to the inductive stimulus of chordomesodome. Therefore, isolated ectoderm unexposed to neural induction and ectoderm transplanted too late differentiate into epidermis only. Late neurula epidermis, no longer convertible into neural tissue, becomes competent to respond to other inductors under the influence of eye vesicle, hindbrain and forebrain respectively. It differentiates into lens, ear vesicle and nasal peats during post-neurular stage of development. Thus, competence is a time-limited phenomenon with a beginning and an ending. As the age of the embryo advances, the competence of the various structures gets gradually reduced. We would start with induction process in the chordates. The primary organizer was first noticed in urodel amphibians. It was found that dorsal lip of the blastopore and the roof of the archenteron of lower chordates and other vertebrates have the same function in the development. In cyclostomes, especially in lampreys, the dorsal lip of the blastopore, when transplanted into the blastocele of another young gastrula, induces the development of a secondary embryo. In bony fishes, induction of secondary embryos were formed by grafting the posterior border of the blastodisc into the blastocele of another embryo. In frog, the formation of a secondary embryo can be induced by the transplantation of the dorsal lip of the blastopore into the blastocele of a young gastrula. In reptiles, archenteron has the same inducing activity as in other vertebrates. In the birds, the anterior half of the primitive streak was established to be the inducing part. Eye development. Eye development presents a typical pattern of series of inductive events. The chordomesoderm, a primary organizer, induces the demarcation of the forebrain and the optic area within it. The development of eyes starts with the evagination of a pair of sac-like optic vesicles from the lateral walls of the forebrain. The external surface of the optic vesicle now compresses out and invaginates inside, so that the vesicle is changed into a dual-walled cup-like structure, the optic cup. The optic cup then serves as a secondary organizer, inducing the structure of the lens. Since the optic vesicle is transforming into an optic cup, the somatic ectoderm overlying the cup undergoes a thickening to form a structure known as lens placard. The placard then invaginates inside and finally pinches away from the parent ectoderm to form a lens vesicle lying in the pupil. The lens substituting together with the retina work as a tertiary organizer of the cornea. The layer of mesenchyme left at the front of the anterior chamber of eye joins with the overlying somatic ectoderm to produce the cornea. Thus, it becomes evident from the foregoing account in the induction sequence that one tissue supplies the stimulus for the development of the other tissue. Since each structure is induced, it can further induce additional structures. The waves of induction could further lead to arranged, accurately timed, total and plant embryo of the animal. We will discuss various mechanisms of neural induction. Development of the ectoderm overlying the roof of the archenteron into neural tissue suggests a direct action upon the ectodermal cells, either by surface interaction or by chemical mediation. One of the broad possibilities is surface interaction of the cells at the inductive interface. The contact of the two cellular layers may provide a device whereby the structural pattern or geometry or behavior of the ectodermal cell membranes is altered directly by the underlying chorda mesodermal cells. Thus, the spatial configuration of the later membranes might induce a change in the spatial configuration of the ectodermal cell membranes. This in turn producing in the interior of the cell changes that determine its development into neural plate. Another broad possibility is a chemical mediation of the inductive effect. Therefore, a chemical substance produced and released by inducing chorda mesoderm cells at the archenteron ectoderm interface may act upon or enter the ectodermal cells to initiate cellular activities leading to neural development. From the experiments done later on, it was suggested that both of these possibilities are very much involved for neural induction. Chemical basis of neural induction. It was found that many different tissues, embryonic or adult, live or dead, from a great variety of species were capable of inducing nervous tissue in amphibian embryos. Moreover, some foreign tissues were found to be much more potent inductors after they had been killed by heat, alcohol or petrol ether treatment, or by freezing or desiccation. The organizer was then implanted into a living embryo in an appropriate stage of development, and it was found that a killed organizer can still induce. Few inorganic agents such as iodine and kaolin, local injury, exposure to saline solutions of excessively high or low pH cause neural differentiation in ectoderm. Artificial inducers such as solvents, acids and chemical dyes are employed for induction studies to understand the chemical nature of the inductors. Different chemical substances of dorsal lip or chorda mesoderm are separated by different biochemical methods to find out the molecule which causes the neural induction, and then the inductive capacity of each molecule was tested separately. Few experiments show that inducing substance or evocator is a protein. Exhaustive attempts were made by different embryologists to understand the real mechanism of neural induction. Some theories have been put forward to understand the mechanism of neural induction, out of which the most important are discussed here. Protein denaturation theory of neural induction. According to Ramsey, neural induction and notochord formation are related to protein denaturation. Site of notochord formation is an amphibian gray crescent, which is a center of high metabolic activity. Such centers of greater metabolic activity correspond to sites of protein denaturation. Gradient theory of neural induction. Toivonen and Yamada stated that two chemically distinct factors are involved in the action of primary inductor. Out of these two factors, one is neuralizing agent and the other is mesodermalizing agent. Regional specificity of the embryonic axis arises from the interaction between two gradients. Neuralizing principle has its highest concentration in the dorsal side of the embryo and diminishes laterally, while the mesodermalizing principle is present as an anterior-posterior gradient with its peak in the posterior region. One factor hypothesis of neural induction. Nikoop, using living notochord as the inductor, postulated that only one factor which first evokes ectoderm to form neural tissue and later causes ectoderm to transform into more posterior and mesodermal structure that are involved. Ionic theory of neural induction. According to Barth and Barth, The actual process of induction may be initiated by release of ions from bound form, representing a change in the ratio between bound to free ions within the cell of early gastrula. Induction of nerve and pigment cells in small aggregates of prospective epidermis of the frog gastrula were found to be dependent on the concentration of the sodium ions. Normal induction of nerve and pigment cells by mesoderm in small implants from the dorsal lip and lateral marginal zones of the early gastrula is dependent on the external concentration of sodium. Genic basis of neural induction. There are evidences that the component tissues of neural inductor becomes differentiated prior to ectodermal cells. During this process, the rate of transcription of mRNA and differential activation of genes becomes manifold, while the differentiation of ectodermal cells is set in only after mid-gastrulation. The mRNA by transcription from the DNA is required for translation of a specific protein for induction. Thus, at the end, we summarize the topic. Organizers are recognized as tissues that influence and organize other tissues to differentiate and produce a tissue or structure that in normal course should not have been formed. Spearman's organizer is a dorsal lip of blastopore of urodel amphibians. If it is transplanted autoplastically, heteroplastically, homoplastically and xenoplastically in any other location of the recipient embryo, a secondary embryo develops. It influences the other tissue, which again has power to induce the another. Thus, the first one is called primary organizer, the second one is called secondary organizer, the third one as tertiary organizer, etc. The influence that comes from the organizer is known as embryonic induction. The chemical basis of this induction is analyzed and finally revealed that these are related to nucleic acids. In other animals, chordal mesoderm in mammals, blastodisc in fishes, and the anterior parts of the primitive strix in birds act as primary organizer to induce neural cells as well as secondary embryo. The competence of the ectodermal cells reduces as age of the embryo advances. Thank you.